All right, class, welcome to class tonight. This Thursday, we'll be going over chapter 16, which is blood, but we'll probably kind of divide this chapter into two parts. So the recording that you're listening to now will kind of be part one, and then I'll post another recording by tomorrow or Friday night, because this weekend you'll have an open note, open book quiz on Sunday. That will be on chapters 15 and 16. And then you also have another Labster lab due, which will be on diabetes on Sunday. So those are your two assignments due for this Sunday. So blood is our kind of introduction to the circulatory system. We'll move on to the heart and blood vessels after this. Blood is our life-sustaining transport vehicle of the cardiovascular system. It works to transport many different types of molecules and chemicals, oxygen, carbon dioxide. It helps to regulate a lot of different ion levels, blood pressure, blood volume, regulating pH levels, and it helps to protect because our white blood cells travel through the blood and they are our body's immune system. So the transport function include delivering oxygen and nutrients to body cells. So we'll look specifically how red blood cells transport oxygen, transporting metabolic wastes to your lungs and kidneys for elimination. So for example, carbon dioxide to your lungs so we can exhale that waste. And our kidneys will get rid of urea or uric acid. And when it talks about metabolic wastes, those are wastes that are coming from metabolic respiration. And that is the idea that your cells are taking in glucose as a food source and creating energy. And carbon dioxide is always a waste product of that chemical reaction. And then transporting hormones, which we talked about in chapter 15. The regulation functions include maintaining body temperature, maintaining pH, maintaining adequate fluid volume. And then the protection functions include preventing blood loss by the ability of your plasma proteins and platelets to initiate a clot formation when a blood vessel is damaged, and also preventing infection. We have agents of immunity that are carried in the plasma portion of blood, like antibodies, complement proteins, and white blood cells. So these are just general functions of blood. It's the only fluid tissue in the body, so it's one of our types of connective tissue. The matrix is non-living, and that's what plasma is. And then cells are the living blood cells, and those are called the formed elements. The formed elements are suspended in the plasma, and these are our formed elements. Erythrocytes are red blood cells, leukocytes are white blood cells, and platelets. So if we were to take a blood sample out from someone, and I just had a blood sample taken from me, and as they centrifuge it down, we can, the blood is spun down, the layers divide into three parts or three layers. Erythrocytes sink to the bottom because they're the densest. So these are the red blood cells. And a hematocrit is the percent of blood volume that is red blood cells. And normally it's a little less in females than it is in males for some reason. White blood cells and platelets make up the buffy coat, which is the thin whitish layer between the red blood cells and the plasma layer that is on top. Plasma layer is on top because it is mainly made of water, so it'll float to the top of that sample. So here is an example of the major components of blood after withdrawing a blood sample, centrifuging it down and seeing how the different layers of blood will separate out into their layers. Again, the erythrocytes are the densest, so they sink to the bottom. The buffy coat, which is just a really thin, lightish color layer, is where our leukocytes are, our white blood cells, and the platelets are, which help with blood clots. So if you have a really large buffy coat, when you look at someone's blood sample, that person probably has an increased number of leukocytes or platelets. And if it's an increased number of leukocytes, that probably means that that person is fighting off a bacterial infection. And then the plasma is the non-living component of blood. It's mostly water. So it's least dense, and that'll always float to the top. So blood is a sticky, opaque fluid. It has metallic taste. I'm sure we've all tasted blood if you've bitten your lip or your cheek. The color will vary with oxygen content. So if it's high in oxygen, it'll be a scarlet red. And if it's low in oxygen, it'll be a darker red. The pH is right around 7.4, the range, and it makes up about 8% of our body weight. And again, the average volume of blood differs a little bit between males and females. Blood plasma, and as I'm working through these slides rather quickly, focus on the parts that I've highlighted. So I'll spend more time on those highlighted parts. So plasma is the straw-colored sticky fluid. It's mostly water, but there's about 100 other dissolved solutes in your plasma, which I don't think I ask you specifics about what makes up plasma. One thing that would be good to know is that albumin is the most prevalent or common plasma protein, and it just functions as a carrier of other molecules, a blood buffer, and contributes to the plasma osmotic pressure. So here's a composition of plasma. Again, it's mostly water. Albumin is the most common plasma protein. The formed elements then are your red blood cells, erythrocytes, white blood cells, leukocytes, and the platelets. Only white blood cells are complete cells, and that's because your red blood cells have no nuclei. They lose their nucleus during maturation, development, and platelets are just cell fragments. So platelets are formed from a larger cell that just kind of loses pieces, and those are the platelets. Most of your formed elements survive in the bloodstream only for a few days, and most of all of your blood cells originate in the bone marrow and never divide. So here's just a look at blood cells underneath a microscope. The red blood cells will be kind of these biconcave reddish cells with no nucleus. The leukocytes will be more large, whiter colored, and then the platelets are the smaller kind of cell fragments that help with forming a blood clot. And here's a look underneath a microscope. Most of what you're looking at is erythrocytes. Any other cell type that's kind of darker stained with a multi-lobed nucleus or a single nucleus that takes up the whole area are one of 5 different types of leukocytes. And then the little dots are platelets. And you will need to know eventually, and we'll go through this, what each leukocyte looks like. You might have learned that in anatomy. Depending on who your teacher was, some people focus on that. So erythrocytes, we're going to talk a little bit about how they transport oxygen. They're a small diameter cell, and it contributes to gas transport. It's a biconcave disc shape. So think of it like a donut, but it still has the middle part in it. It's just very thin. It's anucleate, no nucleus, and it essentially has no organelles because it's filled with one type of protein, hemoglobin, because its only purpose is to transport oxygen. And hemoglobin is the protein that transports oxygen and carbon dioxide for gas transport. The red blood cell diameters are larger than some capillaries, but most can just fit through capillaries by going one at a time. They contain the plasma membrane protein spectrin and some other proteins, which give them a little flexibility to their shape. They are very efficient for gas transport because they have this biconcave shape, which offers a huge surface area relative to its volume for the gas exchange, meaning that because there's just such a large surface area on a red blood cell, it's very easy for oxygen to leave it and carbon dioxide to enter. So that surface area is helpful for gas moving in and out of the cell. Hemoglobin makes up about 97%. There's no mitochondria. They produce ATP anaerobically so that they never consume their own oxygen that they're trying to transport to the tissues. So here's a look at the red blood cell structure. We talked about they're dedicated to respiratory gas transport. Hemoglobin binds reversibly with oxygen, meaning it's able to bind to oxygen and then it's able to let go of it. That's what reversibly means. Hemoglobin contains a red heme pigment bounded to the protein globin, and globin is composed of 4 polypeptide chains, 2 alpha and 2 beta. You don't really need to know that, but you just need to know that the globin has these 4 chains to them, and that each heme pigment is bound to a central globin chain, so that each heme central iron, so iron is at the center of each heme group, and it binds to 1 oxygen. So because of that, so if you look here, this is the structure of hemoglobin. Here are the beta globin chains. So you have 2 beta chains at the top, 2 globin chains at the bottom, they each have a heme group in the center with the green iron molecule, so that each kind of heme group transports one oxygen. So one hemoglobin can transport 4 oxygen molecules because of the 4 chains, the 4 heme groups attached to each 4. So here we have kind of the structure of this heme group. It contains the iron in the middle. That's not super important to know, but you'll see later when we recycle our red blood cells that they recycle the iron, they recycle the heme group. So this is what you do need to know. Each hemoglobin molecule can transport 4 oxygen molecules, and each red blood cell contains 250 million hemoglobin molecules. So let's do a little math here. Then one red blood cell can transport, what is that? 1 trillion oxygen molecules. And that's just one red blood cell can transport 1 trillion oxygen molecules. Oxygen loading in lungs produces oxyhemoglobin. Oxygen unloading produces deoxyhemoglobin, and carbon dioxide loading in tissues produces carbaminohemoglobin. Just letting a couple more people in here. The production of erythrocytes, there might just be one question about how they all come from the same stem cell, but hematopoiesis is the idea of forming red blood cells. It happens in the red bone marrow. Hematopoietic stem cells is just the stem cell that gives rise to all the formed elements. Knowing the exact process and stages of erythropoiesis isn't super important. You just knowing what the word erythropoiesis means, the formation of forming red blood cells. It takes about 15 days. And the important thing is that all your cells, no matter if it's a white blood cell, red blood cell, it all begins with a hematopoietic stem cell. But you guys don't need to know necessarily the stages of erythropoiesis. What I do want to show you is this is what that stem cell looks like. They're also called hematocytoblasts. And I think we even have a different picture that shows how it then goes on to form or differentiate all the different types of cells in general. But this just shows the formation of a red blood cell. You can see how eventually it loses its nucleus. So the nucleus gets ejected, again, to leave as much space in the red blood cell for hemoglobin, so it can transport that 1 trillion oxygen molecule. So one hemoglobin, again, transports 4 oxygen. A couple of regulation and requirements for erythropoiesis. If you have too few red blood cells, that can lead to tissue hypoxia, meaning too little oxygen is delivered to your tissue. Too many red blood cells can increase blood viscosity, meaning it'll make it stickier and thicker. So you really need to regulate the number of red blood cells that are in your blood. More than 2 million red blood cells are made per second, and the balance between red blood cell production and destruction depends on hormones and dietary requirements as well. The big hormone that stimulates the formation of red blood cells is erythropoietin. That'll be released by the kidneys. So this is actually a hormone that's released by your kidneys in response to hypoxia, which is just low oxygen levels. So if your body realizes that not enough oxygen is getting to your tissues, then your kidneys will release this hormone erythropoietin, which will start the formation of red blood cells. So that's all you kind of need to know about that. So I'm going to kind of go through that a little bit. This just shows a normal homeostasis, normal blood oxygen levels. And if we get an imbalance like hypoxia, then the kidney will release that erythropoietin, which will stimulate the red bone marrow to produce red blood cells. I include this homeostatic imbalance in here because some athletes abuse artificial erythropoietin. The use of erythropoietin increases the hematocrit, or your red blood cell count, which allows athletes to increase stamina and the performance, because more red blood cells will deliver, or supposedly deliver, more oxygen to more of your tissues. However, it can increase the hematocrit from 45% up to 65%. And with dehydration, it can concentrate the blood even more. So the blood can become like sludge, because remember the viscosity, the stickiness will increase because there's more red blood cells traveling through. And this can cause clot, stroke, or heart failure. So stay away from artificial erythropoietin. Dietary requirements for erythropoiesis, so getting enough iron, amino acids, lipid, carbohydrates, vitamin B12 and folic acid are necessary. So not necessarily need to know some of those dietary requirements. A little bit about how then we recycle red blood cells. So they live about 100 to 120 days, and then they become kind of recycled in your spleen. So old red blood cells become fragile, hemoglobin starts to degenerate. They usually go to the spleen, which helps to recycle them. This is the red blood cell then breakdown. It breaks it down into its heme, iron, and globin components of that hemoglobin. The iron binds to ferritin and is stored for reuse. So iron will be reused to make more red blood cells. But this is kind of what I think you might have a test question on. The heme itself will be degraded to a yellow pigment called bilirubin, and then the liver will secrete bilirubin as bile into your intestines, where it will be degraded to form urobilirubin that will eventually leave the body in feces. So that's kind of partly how we get rid of some of the parts of our red blood cells. So this kind of takes you through the life cycle of a red blood cell. Some disorders with blood. So there it will be, I think, one matching question about the disorders of blood. And there's a lot of different types of anemia, and you don't need to know all of them. But most of our erythrocyte disorders are classified either as anemia or polycythemia. So anemia is low oxygen carrying capacity, and polycythemia means many. So that'll be the opposite. So we're going to go over a couple different types of anemia, and then we'll look at polycythemia. And we'll go over other disorders with blood in the body that aren't just these two, but having a good understanding of these two will help. So again, anemia, or being anemic, your blood has an abnormally low oxygen carrying capacity. It's usually the sign of the problem rather than the disease itself. So meaning that the problem is just that there's low oxygen carrying capacity. The symptoms can be fatigue, pallor, dyspnea, and chills. And there's three groups based on the cause. So blood loss, so someone can be anemic if they lose a lot of blood, if they're not producing enough red blood cells, or if too many of their red blood cells are being destroyed. So blood loss, if you're hemorrhaging, if you're chronically hemorrhaging, such as from a hemorrhoid or a bleeding ulcer, not enough red blood cells are being produced. Usually this is because you're iron deficient, because you need iron to produce red blood cells. So if you're not getting enough iron, then you won't be able to produce enough red blood cells. Treatment would just be iron supplements for that. And then pernicious anemia, one thing about pernicious anemia, it's an autoimmune disease. So this is something you can't help. It's your body attacking or destroying your own stomach mucosa that produces intrinsic factor. And without intrinsic factor, your stomach can't absorb vitamin B12. And B12 is what is needed to help your red blood cells to divide. So with pernicious anemia, you can usually take low dietary intake or injections of B12. Renal anemia, renal has to do with kidneys. So if you have renal anemia, that means that your kidneys are not producing erythropoietin, which is the hormone that goes on to produce red blood cells. The third kind of group of anemia was not enough red blood cells being produced. So we have aplastic anemia, where they are destroyed or inhibited. So the red bone marrow is destroyed, or something is being inhibiting the red blood cell production. This can be caused by drugs or chemicals. No one really knows this or how, but all formed elements can be affected. So meaning that your platelet number, the clotting factors, will be affected, as well as some immunity defects or your white blood cells. And again, this is a lot of detail about aplastic anemia that you do not need to know. Too many red blood cells produced. This can be just lysis of red blood cells if they're being destroyed. Usually this happens if you have a bad blood transfusion or some other type of infection. hemoglobin abnormalities. We can have sickle cell anemia. So thalassemias, I don't even think I asked you a question about that, but this is just a type of anemia where the red blood cells are being destroyed. And then sickle cell anemia, I don't know specifically, I asked you a question about this, but I include it because in your future medical careers, you might come across this. And I just want you to be aware that at least you've heard it before, even if you don't remember what it is. But basically, it has a mutated hemoglobin. The red blood cells become crescent shaped, so they look sick, and they just can't carry as much oxygen as possible. So it results in poor oxygen delivery as well as even pain because the misshapen red blood cells can block small vessels. One possible benefit that's just a little interesting is that people with sickle cell do not contract malaria, which is kind of interesting. It's also, for some reason, prevalent in Black people of African malaria belt, and their descendants, for some reason. It's a genetic disease that will be passed on. And you can read through those treatments. So this is just a normal red blood cell and what sickle cell anemia looks like. And the difference between having a normal red blood cell and a sickle cell is just one mutation of an amino acid. So that's sickle cell anemia, again, a type of anemia where the red blood cells are being destroyed. And then polycythemia is an abnormal excess of red blood cells. So this will cause viscosity, sludge, sluggish blood flow. And I'm going to kind of pass over that. That's all you need to know. Polycythemia is an abnormal excess of red blood cells. Again, blood doping comes in here where athletes will remove, store, and then reinfuse their red blood cells before an event to increase oxygen levels for stamina. And again, dangerous because you don't want to make your blood nice and sludgy and with high viscosity, so you form clots. Okay, so then leukocytes are our white blood cells. They're the only formed element that it has a complete nucleus and organelles. They make up about 1% of the total blood volume. And if you remember, the white blood cells are always found in that buffy layer, the buffy coat. They can leave capillaries by a process called diapedesis. They move through tissues by kind of a strange amyloidy motion. I don't really need to know that. What you should know, so leukocytosis is when you have a white blood cell count over 11,000 per microliter. And leukocytes in general will increase in response to an infection. So this is very important to know. You know, people get a white blood cell count taken and their leukocytes count are very, very high. That means their body is most likely fighting off some sort of infection, bacterial infection. If their leukocytes are very low, that means they're also bad because that person, you don't want to get that person sick because they don't have their body's defense mechanisms to fight it off. We divide leukocytes into granulocytes and agranulocytes. The granulocytes contain visible cytoplasmic kind of pieces or granules in them. We have 3 of those: neutrophils, eosinophils, basophils, and then the agranulocytes do not contain those visible cytoplasmic granules, and those are lymphocytes and monocytes. Again, here are leukocytes found in the buffy coat of our formed element kind of white blood sample taken. And this in general gives you the percentage of white blood cells found. And neutrophils will always be the most common. And we'll go through, so these 5 white blood cells, you should know what they're kind of easy, and this would be a great thing to do to just make flashcards on. So write the 5 white blood cells, know if it's a granulocyte or an agranulocyte, and then know briefly what each looks like and what each function is and how common it is in the blood. So I know that's a lot of information, but you will have questions on knowing well these 5 white blood cells, what they look like. So maybe make a flashcard, draw the picture of it, test yourself to make sure you can recognize the picture, and then a couple key parts of what each does. And that's what we're going to go over now. So these 5 leukocytes. So neutrophils, you have 2 shown here. They have a multi-lobed nucleus. And again, I think, your test questions will be in black and white because, these look, leukocytes aren't really purple and dark blue. They're just stained this way because of the dye that has been used to make this microscope slide. So you should know in general what makes a neutrophil unique. It has a multi-lobed nucleus. There's 1, 2, 3, 4 lobes to these nuclei. And they do have granules in them. And these first 3 are the granulocytes. You can see those tiny cytoplasmic kind of pieces. An eosinophil just has a bilobed nucleus, so 2 lobes. A basophil also has a bilobed nucleus with cytoplasmic granules, but it's not as easy to see the lobes. A lymphocyte has a small spherical nucleus that kind of fills up the whole cell itself. And then the monocyte has a kidney-shaped nucleus with no granules either. So here are the granulocytes. We'll go through kind of each individually now. So, go through, get out your flashcard. Neutrophil, most numerous white blood cell. You should know that. It's about twice the size of a red blood cell. It has granules. It contains either hydrolytic enzymes or antimicrobial proteins. And the nucleus is lobular because it has multi-lobes to it. So that's the neutrophil. Very phagocytic, meaning it eats things up. It's referred to as a bacterial slayer. It kills microbes by a process called a respiratory burst. And it has defense granules which merge with a phagosome to form like a spear that pierces the holes of an ingested microbe. So that's a neutrophil, and that's what it looks like. An eosinophil is a little less common. It has 2 lobes. The red staining granules contain digestive enzymes, which release enzymes on large parasitic worms digesting their surface, if you ever ingest a worm. They also play a role in allergies and asthma, as well as immune response modulars. So that's an eosinophil, and that's what it looks like. So you have a bilobed nucleus, 2 lobes to it. A basophil, so right away, this is the rarest of the red blood cell. The big thing about basophils is it releases histamine, which is an inflammatory chemical that acts as a vasodilator. So what histamine will do, so basophils will go to an area of infection, release histamine, and vasodilation from the histamine will cause increased blood flow to that inflamed site, which will bring more white blood cells to that area. Functionally similar to mast cells, but the big thing again, basophils are the rarest and they secrete histamine. And that's what a basophil looks like. So then we'll go to our agranulocytes. The 2 are lymphocytes and monocytes. The lymphocytes, after neutrophils, are our second most numerous. They have a large dark purple circular nuclei that almost fills the entire cell. They're crucial to immunity because they will develop T lymphocytes and B lymphocytes. So lymphocytes are kind of our big kind of heavy hitters that help with the immune response because they will go on, we have 2 types of lymphocytes. T cells will act against virus infected cells and tumors. And our B cells is what will give rise to plasma cells, which produce antibodies. So again, just picking out kind of the main function of each leukocyte will be important for this. And no granules in the leukocyte, and the nucleus fills up almost the entire cell. The monocyte is the largest of all the leukocyte. It leaves circulation, it enters tissues, and it becomes a macrophage, which is just an actively phagocytic cell, which is very crucial against all viruses, intracellular bacterial parasites, and chronic infections. It will work to activate other lymphocytes to mount an immune response. And that's what a monocyte is. So it's the largest, and it has more of a kidney bean shaped nucleus. So again, understanding what the leukocytes look like, how common they are, or knowing like what's the most numerous, what's the least numerous, what's the largest, the monocyte, and then knowing just a really brief, like general function of each will be important. Leukopoiesis is the production of white blood cells, needing interleukins, collicle stimulating factors. I don't think I asked you specifics about production of leukocytes, but similarly, they arise from kind of one stem cell line. The lymphocytes will go on to form the T lymphocytes and the B lymphocytes, those T and B cells. So again, this is the picture that I was trying to show before, but this is the hematocytoblast, which again is the common hematopoietic stem cell. where all cells will originate from. So then they'll differentiate into their appropriate leukocyte, as shown there. So that's the hematocytoblast, starting with one stem cell, and then they'll go on to differentiate. And from there, you don't really need to know the process by how they're formed. Many hematopoietic hormones are used clinically to stimulate bone marrow of cancer patients, for example, receiving chemotherapy. So for example, cancer patients who are losing a lot of their cells, there's hormones that can try to stimulate the production of more cells, also used to protect the immune response of AIDS patients. Some leukocyte disorders, so again, more disorders of blood, we have leukemias and infectious mononucleosis. A leukopenia is an abnormally low white blood cell count. It can be drug-induced, particularly by an anti-cancer drug, because anti-cancer drugs will try to kill off all the cells, and when they do that, they kill off the good cells too. So leukopenia is low white blood cell count. Leukemia is a cancerous condition involving an overproduction of an abnormal white blood cell. So leukemia is cancer of white blood cells. There's an abnormal production, or there's an overproduction of abnormal white blood cells. So that is a leukemia. It can be acute or chronic. So that is leukemia, cancerous. Without treatments, all leukemias are fatal. Death is usually from an internal hemorrhage. The treatments usually include irradiation, anti-leukemic drugs, and stem cell transplants. So that's leukemia. Infectious mononucleosis is a highly contagious virus disease, the kissing disease of mono. It's caused by Epstein-Barr virus. The symptoms are tired, achy, chronic sore throat, low fever, and it can run its course with rest in 4 to 6 weeks. I don't know if I asked you about that one. Really simply, I include platelets in our discussion because they're part of the formed elements of blood. I don't know if there's any platelet-specific questions on your exam, but I want you to know that, well, I guess there will, because we talk a lot about clotting, but platelets are fragments of a larger megakaryocyte. So basically, we have a megakaryocyte that kind of fragments off pieces, and those pieces are platelets. And the big function of platelets is to form a temporary platelet plug that helps to seal any sort of break in a blood vessel. And circulating platelets are kept inactive and mobile by nitric oxide and prostacyclin from endothelial cells lining your blood vessels. So here's platelet formation. You don't need to know that, but again, it's just pieces breaking off of a megakaryoblast. So you can see the platelets forming as pieces coming off of this megakaryocyte, as shown there. This is a summary of the formed elements of blood, maybe a good chart to study as you're working through knowing and learning the white blood cells. Actually, this is a great chart to study because it tells you, I mean, probably just studying this chart, you would probably be good with several questions on the exam too, because it also tells the function of those white blood cells. So yeah, that's good. Hemostasis is the fast series of reactions for the stoppage of bleeding. And I think you might have one question on your lecture exam about what is the first step that happens to try to stop bleeding. And hemostasis requires clotting factors and substances released by platelets and other injured tissues. So the first step is the vascular spasm. Then the platelet plug forms and coagulation forms, which is a blood clot. So the vascular spasm is step one. This is where your vessel responds to injury with vasoconstriction. So there's direct injury, for example, to a vascular smooth muscle cell, chemicals are released, pain reflexes will occur. What happens is it will just significantly reduce blood flow until other mechanisms kick in. So again, we're looking at how to stop bleeding. So hemostasis is reactions to stop bleeding. So this could occur, when you slice your finger, when you're chopping vegetables, if it's even more serious than that or less serious than that. The first step is to reduce blood flow to that area where there was damage. So vascular spasm is the first step. And that just means that the vessels will vasoconstrict or try to just shut off blood flow to try to stop the bleeding from leaving the body. And then a platelet plug forms where the platelets stick to collagen fibers to try to form a plug, to try to just keep blood from draining out as well. When activated, the platelets will swell, become spiked and sticky, release more chemical messengers. This is one of the positive feedback cycles in the body, which means as more platelets stick together, they release more and more chemicals, which cause more platelets to stick together to release more chemicals. The platelet plugs are fine for very small vessel tears, but larger breaks in your vessels, a platelet plug can't form, a plug if you completely, cut off your arm or something. So we need an additional step for larger breaks. Coagulation is blood clotting. This reinforces your platelet plug with fibrin threads. So blood clots are very effective in sealing a larger vessel breaks, maybe not like your arm cut off, that was a bad example, but a larger blood vessel. Blood will be transformed from a liquid to a gel, and a series of reactions using different clotting factors or plasma proteins will occur. And coagulation itself occurs in 3 phases, which I don't even think we go through the 3 phases of coagulation, but this is telling you the events of hemostasis, which is the idea of trying to stop blood flow from an injured vessel. So again, vascular spasm is the first step where smooth muscle contracts, causing vasoconstriction to that area of damage. Platelet plug formation, and then coagulation, which is actually the formation of the clot. Coagulation, the phases, I don't think I asked you questions on, so I'm gonna pass over this for coagulation. I included this in previous semesters because in understanding these phases of coagulation, we talk about then anticoagulants, because many of you probably know people who are on anticoagulants, so anti-clotting measures. And an anticoagulant is just a factor that normally dominates in the blood to inhibit coagulation. And we can take, so this is a look at the scanning electron micrograph. This is actually an erythrocyte trapped in a fibrous mesh of a clot. But again, you don't necessarily need to know those steps of coagulation formation. But I included this in a previous one because many of us might know people who are on a warfarin, or what's the other big anti-clotting factor. My dad is on one, so that's why this is a special place in my heart. So the disorders of hemostasis, you can have 2 major types of disorders: thromboembolic disorders, which result in an undesirable clot formation. So, in this process of trying to stop your blood vessel from bleeding out, you might form clots that are undesirable. And then the bleeding disorder is an abnormality that prevents normal clot formation itself. Some thrombi and emboli. So I want you to know a little bit of differences between a thrombus, an embolus, and then an embolism, because again, these are very common that you might see in your future medical careers, these words. If you go on to nursing school or other school, you'll go over this even more, but I just wanted to give you one chance to at least have heard about these before. So a thrombus is a clot that develops and persists in an unbroken blood vessel. So this could block circulation, leading to tissue death. So a thrombus, so these are clots that form when we don't want them to. And clots in general can form from atherosclerosis, inflammation, slowly flowing blood, or blood stasis from immobility. So that's what a thrombus is. A thrombus is a clot that you don't want. An embolus is a thrombus freely floating in your bloodstream. So now let's say the thrombus starts to travel. Maybe it goes up to your brain, maybe it goes to your heart, maybe it goes to your lungs. But when a thrombus is freely floating in the bloodstream, it's called an embolus. And then an embolism is when an embolus actually obstructs a vessel. For example, in the pulmonary, so in your pulmonary capillaries, arteries, so if you have an embolism in your lungs, or a cerebral embolism in your brain. So why are clots bad? Well, anytime, so if we have a thrombus, it's freely floating. If it actually becomes an embolism and it obstructs the vessel, that means that that area or that tissue part of your body is not getting oxygen. And every part of our body needs oxygen. And we will learn why. It's very exciting. We'll learn why our body actually needs oxygen towards the end of the semester. But if there's a tissue and it's blocked, it cannot get the oxygen, that part of the tissue dies almost immediately, or it scars, it's destroyed, and it can never come back again. In the heart, this will cause a heart attack. In the brain, it could cause a stroke. If this happens in your lungs, well then, I mean, you're not even getting oxygen going from the air you breathe in into the blood. So these three words are very important to know. So I want you to know that. And again, things that can cause this, atherosclerosis is a buildup of plaque in your blood vessels, inflammation, slowly flowing blood. So anytime you might have high blood pressure, it eventually, the blood can become sluggish from a clot, or blood stasis from immobility. So again, look at your own lifestyle. Exercise, diet are very important. Here we get to some anticoagulant drugs used to prevent undesirable clotting. So in people with kind of these preeminent conditions, such as my dad, for example, he had a blood clot in his lungs. He was very overweight, very heavy. He ended up in the hospital. And then he totally changed his diet around. And I think the reason why he's still alive to this day is because he kind of got a wake-up call. But he's on warfarin. So anyway, aspirin can lower heart attack incidence. So these are all anticoagulant drugs that will try to prevent clotting. Heparin and warfarin. So those are some anticoagulant drugs you might have heard of. I promise in a couple minutes we'll be done here. Thrombocytopenia is a deficient number of circulating platelets. So again, this suffix, penia, always means smaller. They're deficient. This is a look at Petitchet. Can occur with the feet. Again, you don't necessarily need to know that. Bleeding disorders, usually due to impaired liver function. Liver disease can also prevent liver from producing bile. So I don't know if I talk too many about that. Hemophilia is a hereditary bleeding disorder. Again, I don't think I asked you guys a specific question on this, but I just want you to have at least heard this name before. So it can be usually due to genetic, always a bleeding disorder having to do with prolonged bleeding, especially into joint cavities. So that's hemophilia. Disseminated intravascular coagulation involves both clotting and severe bleeding, can occur in complications in pregnancy, blood transfusions. So then blood transfusions, and I think this is where we'll stop. So the second part of this recording will be short, but I'm going to do this tomorrow in kind of a separate recording because blood transfusions and blood typing is kind of a whole other topic that I want you to be fresh for before you listen to, because there will be a couple questions about blood typing, blood transfusions, and the like. So I'm going to go ahead and stop the recording. Look for the second half of this. It's just about 10 slides. It won't be long, but posted by Friday night so that you can use that to help with